Okay, welcome to Let's Get to the Marks, going through the AS-Biology Paper 2, 2023, hour and 30 minutes, 10 questions. Let's get into it.
Stick around.
I'm gonna have to science the shit out of this.
Question 1.1. Which statement about the function of ribosomes is correct? Tick one box only. Why don't you pause the video, have a go, and then unpause and watch the walkthrough. Okay, we've got the site of transcription catalyze the joining of amino acids by hydrolysis. Well, transcription happens in the nucleus. It's the formation of mRNA, so it's nothing to do with amino acids. And amino acids wouldn't get joined together by hydrolysis. That would break apart amino acids in a peptide. So that is incorrecto. Site of transcription. That doesn't happen on the ribosomes. Transcription is in the nucleus. Catalyze the joining of nucleotides. No. Nucleotides will get joined together in transcription, but it's not what happens on the ribosomes. And there we go. The only thing they've said there that's true is that the nucleotides are joined together by condensation reactions. But yes, that's untrue. Right, the site of translation, is that what goes on ribosomes? Yes, because transcription happens in the nucleus and translation happens later, hence the LA, happens later on the ribosome. And that is the joining of amino acids by condensation reactions. That is true to form a polypeptide. Right, this is another incorrect one. Site of translation, catalyze the joining of nucleotides. No, the joining of nucleotides happens when we're forming mRNA in transcription. And it's not by hydrolysis, it would be by condensation. So lots of incorrect statements in those three sentences. The correct answer there is the third one. Bash, bish, bash. Right, 1.2, name two biological molecules. This is a really difficult one, actually. Name two biological molecules coded for by a gene that do not include a polypeptide or protein in your answers. So obviously, mRNA, when we're making mRNA in transcription, we know that is coded for by a specific gene, and therefore tRNA also coded for by a specific gene, and rRNA. So you've got all the different forms of RNA. Any of those two would get you a mark. 1.3, scientists investigate the structure of endoplasmic reticulum. Table 1 shows the scientists' results. Type of endoplasmic reticulum. So you've got a rough endoplasmic reticulum. You might remember the rough endoplasmic reticulum. That is a real dodgy drawing. That looks like an alien or something. But yeah, it is studded with ribosomes. And then the smooth endoplasmic reticulum, that looks more like Golgi apparatus. Yeah, I've drawn that like the Golgi apparatus. Anyway, that was crusty. Doesn't have ribosomes. Use the data in table 1 to suggest how the structure of the rough endoplasmic reticulum is different from the structure of the smooth endoplasmic reticulum and how this is related to their function. So we can see the rough endoplasmic reticulum is made up of less phospholipids. So this kind of gets us the marks. Rough endoplasmic reticulum contains less phospholipids. Why? What is it made up more of? Well, it contains more protein as it is covered in ribosomes where protein synthesis is taking place. It's the site of translation, isn't it? So not ribosomes. It's more like ribosomes. Yeah. So rough endoplasmic reticulum. So we would expect there to be less phospholipids because it's the site of protein synthesis. So it's making proteins and it's covered in ribosomes, hence the name rough. Now we're looking at the smooth endoplasmic reticulum. Why would we expect more phospholipids there? We would expect there are more phospholipids. You'd expect more lipids. There are more phospholipids in the smooth endoplasmic reticulum because it is the site of lipid packaging and synthesis. So basically, there's a lot of modification to lipids going on there. So we'd expect phosphate groups to be getting added to lipids there. We'd expect glycolipids to be made there. We'd expect all kinds of modification and packaging of lipids to be going on there. So yeah. So I'd say you're going to get a mark for really talking about there's more protein in the rough endoplasmic reticulum because it contains protein ribosomes that do protein synthesis. So that's going to be 2 marks. And then you're going to get more lipids in the smooth endoplasmic reticulum because it's the site of lipid synthesis or lipid packaging and modification. There we go. Bosh. Excuse my crusty drawings. Oh my days. That's the end of question one. That was quick. 2.1, figure one shows an ATP synthase enzyme in the inner mitochondrial membrane. So here we've got the ATP synthase. It's going through the inner mitochondrial membrane. We've got hydrogen ions all over this. The hydrogen ions coming through, ADP plus PI going to ATP. Nice. Complete the passage with appropriate terms for 2 marks. ATP synthase compromises several polypeptides. So it's said to have, what do we call it when you have more than one polypeptide chain bound together to make a tertiary structure in the protein or the final 3D shape, not the tertiary structure? We call it quaternary. So it is a quaternary uh, structure. It's said to have a quaternary structure. Not all proteins have a quaternary structure. Some just go up to tertiary, which is the final 3D shape. But if you have more than one polypeptide, you end up with a quaternary structure, just like hemoglobin has 4 polypeptides. Enough waffle. It catalyzes the synthesis of an ATP molecule by a something reaction. Okay, so what do we call reactions that join together smaller monomers or small molecules, not necessarily monomers, into slightly larger molecules? We call them condensation reactions. They're addition reactions. So it's talking about synthesis. You might have wanted to put hydrolysis here, but you should look at that word synthesis, say, oh, that's making ATP. It's asking me what reaction makes ATP. The making reaction in biology is condensation. The breaking reaction is hydrolysis. This involves the something of a water molecule. I guess there's options here: elimination, removal, loss. Any of those um, would be good. The production of a water molecule, the elimination, the loss, or the removal of a water molecule. The ATP synthase in figure one is in a mitochondrion. So it's in a mitochondria. So it would catalyze reactions during what reaction happens in the mitochondria? It's going to be respiration. You could say aerobic respiration because the mitochondria doesn't do anaerobic, so it has to be aerobic. You might want to put that. Right, quaternary, condensation, elimination, loss or removal, and respiration. Nice. Okay, 2.2, as shown in figure one, ATP synthase has 2 functions. It catalyzes the synthesis of ATP. It allows the movement of hydrogen ions. So just how the shape of the ATP synthase allows it to have these 2 functions. Explain your answer. How can it catalyze the synthesis of ATP? That means it has a um, tertiary structure or quaternary structure. We already established that. It has a tertiary structure or 3D shape. Um, oh, let's go back. Let's say which bit of it has the unique shape. Its active site has a specific 3D shape. I started talking about tertiary structure, but of course we just confirmed that it has a quaternary earlier in an earlier part, so I don't want to waffle about that. Its active site has a specific 3D shape that is complementary to the substrate. When it's making ATP, what's the substrate? So it's complementary to ADP plus PI, forms an enzyme substrate complex. So that's how its shape allows it to catalyze the formation of ATP because its active site has a specific 3D shape or tertiary structure, quaternary structure, whatever, that is complementary to, oh my gosh, AD. I think I've got AD, whatever that is. Um, ADP plus PI forms an enzyme substrate complex. Good. Right, that's going to get us at least 2 marks. I'm going to have to delete that because I'm going to scroll down. Now we need to talk about how does the shape of the ATP synthase allow it to move the hydrogen ions? Well, we've already established that the ATP synthase has a uniquely shaped active site that is complementary to the ADP and PI, the 3D structure of this active site, so that they can be catalyzed by joining in there. But I would suggest that also, well, let's not redraw it out. Also, it has a channel, which it kind of showed in the diagram, through which the hydrogen ions can move through. So I would say it has a channel through the protein that allows hydrogen ions to move through by, what do we call it when you diffuse through a protein? By facilitated diffusion. So that is what's going to get the other 2 marks. Basically, it's got a channel, or we could say it's specific 3D shape that allows hydrogen ions to move through it by facilitated diffusion. Bosh. Okay, that's probably bad to talk about the specific 3D shape, but there we go. All right, let's move on. Don't want to spend forever doing these questions. Galacto olig What's galacto-oligosaccharides? GOS. Thank you. We'll use the abbreviation. GOS are polymers of galactose. So lots of galactose molecules joined together. Explain why GOS are described as polysaccharides. Well, they've just told us, yeah. So they are made up of many monomers of galactose joined together by condensation reaction. I'm writing everything I can about a polysaccharide. So a polysaccharide is monomers of a sugar. In this case, we want to be specific, it's galactose, joined together by condensation reactions. What do we call the bonds? Let's mention the bonds. Held together, or I should say the monomers are held together by glycosidic bonds or links, whatever you want to say. That's going to easily be enough for 2 marks. I've kind of said 3 things. So they're monomers made up of galactose monomers. That's why it's a poly. By condensation and glycosidic bonds. I've said 3 things there. That's enough. Let's move on, Tom. Give 2 differences between the GOS structure and the lactose structure. So GOS is this galactose polymer. So, oh, I'm not even going to draw hexagons, but you get the idea, right? It's like loads and loads and loads. And what is the monomer in it? It's made of, that's GOS. And GOS is made of galactose, right? Galactios. And lactose is a disaccharide, right? Yeah, lots and lots of bonds in galactose. Lactose is made of, is it just made of 2 galactose? No, it's not. It's made of a galactose and an alpha glucose. So we've got some differences. Let's go for it. Right, lactose is made of alpha glucose and galactose, whereas GOS only contains galactose. Ta-da! There's one difference right there. And then this has got, GOS has many glycosidic bonds. GOS has many glycosidic bonds, whereas lactose only has one. I reckon, yeah, it's made of many monomers, many molecules joined together, lots of condensation reactions, trying to think what else, what else, what else. No alpha glucose, we said that. This is a disaccharide, this is a polysaccharide. That's just a name. That probably won't, that's not really the structure, is it? Just saying it's a di and a poly, but that's made of 2 monomers. This is made of many monomers. Those are different monomers. We already mentioned that. Okay, let's move on. Scraping out the bottom of the bowl there, trying to find some extra points. Explain why amylase produced in the human digestive system does not digest GOS. So this is going to be quite simple. If this is amylase, right? They're basically like, what does amylase normally digest? Well, it's either amylopectin or amylose. So it's going to do starch, which can come in the form of amylopectin or amylose. So it means that the active site of the amylase is complementary to starch, forms an ES complex with starch, but cannot form an ES complex with GOS, as GOS has a different 3D shape. Bosh! Need to put that into words. So amylase, oh my days. Amylase has an active site with a complementary 3D shape, I don't know, tertiary structure. I don't even know if it has quaternary or whatever, but has a complementary 3D shape tertiary structure to the starch substrate, forms ES complex, but cannot do this with GOS as it has a different shape. I'm longing this out, people, not complementary to the active site of amylase. Oh my God, I've longed that out. So it can't form an enzyme substrate complex with amylase. I longed that out, that explanation. I'm sure I could have written that in just one sentence. I could have just said the GOS has a different shape to starch. GOS is not complementary to the active site of amylase. GOS cannot form an enzyme substrate complex. And that would get you the 2 marks. Don't know why I wrote a whole English 25-mark essay on a 2-mark question there, but here we go. Right, 3.4. Prebiotics are foods used to promote good health in humans. I'm sure we call them probiotics, but there we go. Right. Prebiotics stimulate the growth of healthy bacterial populations in the human digestive system. You know them little yogurt drinks you buy in a supermarket. The bacteria in these healthy populations produce enzymes that hydrolyze GOS. Suggest how GOS can work as a prebiotic. So, what is GOS made of? So, when GOS is hydrolyzed, it releases galactose molecules or monomers. Oh my days, galactose molecules. Now, how will the bacteria use the galactose molecules, and why does this help them, like their population increase? So we want to say the bacteria use the galactose for respiration, um, releases ATP or produces ATP used for metabolic reactions, um, for growth, uh, let's say for binary fission, which is the way that bacteria reproduce, right? So they're using it for metabolic reactions, i.e., they might use the galactose to create ATP so they can do hydrolysis, they can digest stuff, they can essentially do binary fission and increase their population by reproducing. So for binary fission, for reproduction. Whether or not you'd have to check the mark scheme to see if reproduction would get you a mark instead of binary fission, but yeah. So when it's hydrolyzed, it forms galactose, 1 mark, can be used for respiration, to produce ATP for binary fission. That would be my 3 mark at it. All right. Oh my good gosh. Question 4. This question is about how the flow of blood into and through the heart. This question is about the flow of blood into and through the heart. Add numbers 1 to 6 to table 2 to give the order of structures through which blood will pass as it enters the heart and flows through the left ventricle. Use each number only once. Number 4 has been done for you. This is pretty easy, but oh my days. Am I really going to do it? Yeah, all right then. Oh my good gosh. Don't really want to draw a pink heart, but what are you going to do? Okay, so here is a blood vessel coming into the left side of the heart. Um, yeah, right. You should all know what that vessel is from that incredible drawing right there. And then I'm just dealing with the left side of the heart here, folks. Oh my days, what is this? This is looking horrendously ugly. Um, yeah, oh my days, what the hell is that? Oh my good gosh. All right. Yeah, I don't know what the hell that is. This is the left side of the heart. So, what blood vessel is this? The blood returns to the heart from the lungs through the PV, right? Am I right? Into the LA. I nearly said R. Remember, Oh my days. Oh geez. LA. Then we got this here, AV valve, although you can call that try before you buy. So this is the bicuspid valve. Then I've got the LV, and then we've got the semi-lunar SL, I'm going to call that semi-lunar. And then up here is the aorta. Okay, let's see if we can number these. So we're returning to the heart and the pulmonary vein. Number one, where is left atrium? Left atrium. Number two, then we want to go through the AV valve, the left atrioventricular valve, otherwise known as the, try before you buy, the bicuspid valve. So three, then we're into the left ventricle. Four, then we're into the semi-lunar valve, left semi-lunar valve. Five, and then I'm looking for aorta peeps. Six. Boom. All right, that's going to get us 2 marks. Lovely jubbly. So always use your sketchy drawings. You should be able to draw a heart out and label it. You can do that while you're watching Netflix. Easy peasy. Okay, we're on to 4.2. So scientists investigated heart activity of humans at rest during medium intensity exercise and during high intensity exercise. So we've got three variables. Well, that we've changed our independent variable to rest, medium intensity, and high intensity exercise. Okay, here are the results. Oh my days, one of these ugly graphs. So we've got the mean volume of the left ventricle on the left. And at rest, this is the volume before it contracts, and this is the volume after it contracts. Probably not going to ask us, but I guess we could work out the difference between the two. That might give us an idea of the volume of blood being released, maybe. We've also got here this shaded gray block. What is that? That is heart rate. Okay, so we've got the volume before contraction, volume after contraction, and heart rate. Now, you might notice that this is one of those biology graphs that has two y-axes. We've got one here on the left, and we've got one on the right. And this can get confusing when reading. You can kind of think, oh, do I read the heart rate off of there and that off there? No, this bar is only to do with the left because this is the volume before contraction, and this here is the volume after contraction. So we read these two bars from here and here. And when we want to analyze this bar, which is the heart rate, we go to this axis, which has the mean heart rate in beats per minute. So that's the way we do it. Let's go down and actually see what's going on. Oh, it looks like there's an increase in the volume of blood pumped out of the heart. Yeah, look at the difference between the starting and final. There's a big increase in the volume being pumped out once we start exercise. What about the heart rate? Yeah, looks like there's an increase in heart rate as well, although something's going on with the volume here. Think, yeah, these are level. So the volume doesn't actually change between medium and high intensity exercise, but the heart rate changes whatever exercise you're doing, whether it's light exercise or harder exercise. Right, let's just get down. Stroke volume is equal to volume of blood leaving a ventricle with each contraction. Cardiac output is the stroke volume. So it's the amount of blood that pumps out your heart each time the left ventricle contracts times the number of contractions per minute, or the heart rate. So the number of beats per minute times the amount of blood going out with each squeeze gives us the total amount of blood going out per minute, and we call that the cardiac output. Use all the information to describe what causes the increase. So they're telling us the cardiac output increases when we go from rest to medium intensity exercise, and when we go from medium intensity to high intensity exercise. Worth 2 marks. You do not need to calculate the cardiac output. Great, so we don't need to do that. Just describe what causes an increase in the cardiac output here and what causes an increase here. Let's go back up to the graph and have a look. Okay, so they told us that cardiac output is equal to the volume times the heart rate. Well, if we look at rest, the heart rate has gone up. That's going to cause an increase in the cardiac output alone. But also check out the difference here. This is the stroke volume, I'm guessing, the difference between the volume before and after. So that's like the stroke volume. And well, you can see that the volume anyway has changed. The stroke volume seems to have gone up as well. So both the heart rate and the stroke volume increase. That's bound to increase the total amount of blood leaving the heart every minute, the cardiac output. So we want to put that answer in here. So both stroke volume and heart rate, whoopsie, I didn't put a space there, never mind. Yeah, both stroke volume and heart rate increase from rest to medium, God, this is long, intensity exercise. And that's going to get us 2 marks. They just ask for a description. They don't want us to explain why. They don't want us to say increased blood flow to the muscles, more oxygen, more aerobic respiration, more ATP for muscle contraction. We don't say any of that because we didn't see the word explain. So make sure you double bubble check the command words. And this was a describe, no explain necessary. Right, let's go and describe the relationship between medium intensity compared to high intensity exercise. What is the change? What causes the change in the cardiac output? Well, this time, if we measure the difference in the volume between before and after contraction of the left ventricle, there's no difference in stroke volume. So the stroke volume does not cause the increase in cardiac output. That has stayed the same. But what has changed, we read over here, is the heart rate has gone up. It looks like it's gone up like 18 beats a minute or something. So the heart rate has gone up. So we just need to put that into words. And they're just looking at, can you analyze a graph? And it's not always easy, actually, but there we go. Right. So, oh my days, that's not good. Oh, what happened there? Do 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 do. Okay, got a bit excited there. Let's put this in. So we want to say medium intensity exercise are high. So no change in stroke volume. So the increase in cardiac output is down to the increase in heart rate alone. According to the results we've got, it's down to, it's not an ion, it's an increase. I always have a spelling mistake going on, don't I? I can't help it. I think I get mixed up with chemistry talking about ions. Okay, there we go. No change in the stroke volume, but there was an increase in the heart rate. So the increase in cardiac output must be down to the increase in heart rate. Good. And that's when we go from medium to high intensity. That was just a description. You don't need to explain it. Name the type of blood vessel that controls blood flow to the muscles and explain these blood vessels, how these blood vessels change blood flow during exercise. So the name of the blood vessel, well, we've got some choices here. There are 5 blood vessels that we really deal with. We have capillary, we have arteries, we have veins, and then new ones for A level, arteriole and venules. So you got 5. Capillaries are 1 cell thick, endothelium, no elastic, no muscle. They cannot change the size of their lumen, okay? And arteries and veins, yeah, they're not really into massively changing the size of their lumen either. If you change the size of an artery's lumen, you're going to affect the blood pressure in the body. That could have disastrous consequences. But arterioles and venules, especially arterioles, these can change the width of their lumen. And it is the arterioles that are supplying the muscles. Obviously, eventually it goes down to capillaries. So you might have put capillaries there, but capillaries cannot change the size of their lumen. So we're not talking about those. So we're talking about arterioles. Yes, arterioles break down to capillaries in the muscle tissue, but yeah, the capillaries cannot alter their lumen size. So why, how, and explain how these blood vessels, right? So we need an explanation of how. They do it by the process of vasodilation. You might remember this from GCSE if you did triple. Vasodilation is the process in which the lumen of the arteriole widens, allowing greater blood flow to the muscles. Now, saying greater blood flow to the muscles is not going to get me a mark. So, yeah, what have we got here? We've got arterioles. Vasodilation is the name of the process. I think that's probably maybe the same marking point as saying the lumen of the arteriole widens. It's like an explanation of vasodilation in case you can't remember that term. And then we want to know the actual science behind that. Well, what type of muscle do you have in blood vessels, in arterioles, or in your small intestine? What's that muscle called? It's smooth muscle or circular muscle. And this muscle must relax. In order to widen the lumen, this muscle relaxes. So that is probably your third mark right there. You won't actually need that, allowing greater blood flow to the muscles, because they didn't ask for why it happens. They said how. So explain how. They didn't say why. And so we don't need to waffle about that. Okay, coolio. Let's move on. Ooh. 5.1, figure 3 shows diagrams of 6 possible arrangements of chromosomes in cells, A, B, C, D, E, F. These look like chromosomes. Yeah, we got homologous pairs, we got single chromatids. Before meiosis, a cell of a rice plant has 12. So before meiosis, it has 12 pairs of homologous chromosomes, 24 chromosomes in total. Okay. Um, Give the letter of the diagram from figure 3 that correctly shows the chromosome content of the rice cells after the first meiotic division and after the second meiotic division. Right, so what you've got to do here, this can be quite confusing, this question, but if we break this sentence down here, 12 pairs of homologous chromosomes. What is a pair of homologous chromosomes? Well, a pair of homologous chromosomes, that is a chromosome made of two sister chromatids. This is a pair of homologous chromosomes. So we're looking for 12 of these pairs. Remember, each one of these X shapes, which is made of two chromatids, is known as one chromosome. So 24 chromosomes in total. I'd have to have 12 X's there and 12 X's there, because 24 X shapes. So before meiosis, it has 24 X shapes. So after meiosis, I'm expecting it to go from 24 chromosomes that look like this. That does not count as two chromosomes. When they are joined together, two chromatids, they count as one chromosome. And they told us they're in homologous pairs. So they're in the X-shaped formation, and there were 24 of them. So after the first division, after you half them once, what will it look like? A, B, C, D, or E? It's going to look like B, because there are 12 there. So after the first meiotic division, we're down to just 12 chromosomes from 24. Yeah, get that? There were 24 X shapes. We've broken down to 12 X shapes. Now, in the second meiotic division, the sister chromatids separate and pull apart away from each other to the different poles of the cell, or yeah. And then essentially we're going to end up with either D, E, or F. So imagine you half this now. What are you going to have? D, E, or F? Well, there's 24 chromatids in there, but it's now going to half into two cells. And sorry, there's 12 chromosomes, but 24 chromatids. So when they separate, it's only going to be 12 chromatids left. So it's going to be E. Got to long that explanation out. Oi. Hi, hi, hi. Scientists have produced a mutated rice variety in which there is no crossing over. So you need to use your highlighter. There's no crossing over. What is crossing over, I hear you say? Crossing over is when the chromosomes line up. Look, there's a little centromere. When the chromosomes line up like that in homologous pairs. Homologous pairs means you have one from mum, one from dad. Let's make the pink one from dad. We're all about colors here. Pink was actually the favorite color of boys during the Victorian era, and blue was the girls' color. Isn't that different? Right, than today. So what happens is the sister chromatids in the homologous pair wrap around each other and swap alleles. We call that a chiasmata, the area where they have joined. Anywho, it's saying that this doesn't happen. A population of mutant rice variety produced by sexual reproduction, that must be important, otherwise they wouldn't have said it, shows genetic variation. So we've got variation by sexual reproduction. Populations of non-mutant rice also show genetic variation. So both the mutant rice that can't do the crossing over, remember the mutants have been made so they can't form a chiasmata, they can't do crossing over. Suggest and explain similarities and differences in the causes of genetic variation within these rice populations. So this is really testing you on your subject knowledge. What are the causes of genetic variation? Which ones will be the same between the mutant rice that can't cross over, and which ones will be like which ones are the same, the causes of variation. Anywho, here are the causes of variation, peeps. You might want to have a go, grab a pen, grab a bit of paper, write down all the causes of variation you know. First clue is the sexual reproduction, right? So the population of mutant rice produced by sexual reproduction shows genetic variation. So sexual reproduction, we have random fusion of gametes, right? We don't know which gametes are going where when it's sexual reproduction. So that is going to be a similarity. Random fusion of gametes in both varieties. I'm guessing both varieties are made by sexual reproduction. Then, so that is a similarity. I'd then say they both undergo independent segregation or random segregation, however we want to say it. So random, yoi, segregation. Oh my gosh, I swear I haven't spelled that right. Or independent random separation, segregate. Oh no, we'll just call it independent segregation. Oh my gosh, that is just foul. I don't even know what I'm writing. Okay, both of these are going to be similarities. They're both going to, we don't know which chromatids, right, are going to, we don't know which chromatids are going to which pole of the cell. It's completely random which chromatids separate in the second mitotic division and end up in which pole of the cell. It's random which of the homologous pairs goes north, goes south, or east and west to the different poles during the first mitotic division. So that is random, and they both still do that. But what we would say in mutant rice, there'll be no chiasmata formed. So there's no chiasmata formed, no swapping alleles, alleles at crossing over. However, in non-mutant rice, there is crossing over. So alleles swapped. My God, I can't spell. Swapped between homologous pairs. So there we go. There's the difference, is that you're going to get chiasmata, you're going to get this thing in the non-mutant rice. You're going to get swapping of alleles between homologous pairs, whereas both of them will do the random fusion of gametes during sexual reproduction and the random or independent segregation. I longed that out, folks. Let's move on. 5.1, 6.1. Describe the hydrolysis reactions involved in the digestion of triglycerides. Do not write about the activity of lipase. Okay, thanks for that. Basically, what are they asking? Describe hydrolysis reactions. That's what they're asking you. And then in the context of triglycerides. So if you wanted to describe a hydrolysis reaction, what would you say? You'd say breaking bonds. Which bond? We're in the context of triglycerides. Breaking ester bonds. Bosh. And what is unique about hydrolysis? What does it use? Using water molecules. Okay, so we're breaking ester bonds using water molecules. And as it's in the context of triglycerides, I'm going to say what's formed. So what are the products, guys? Forming fatty acids and glycerol. I reckon the two best marks are just the first ones, breaking ester bonds using water. You basically said, what are the bonds, context of triglycerides, and you've described that it's a breaking reaction involving water molecules. Anyway, I thought I'd put the products there. 6.2, all mammals produce lipase. Okay, so we've got a lipase called CEL. Okay, CEL digests triglycerides. So it's a lipase that breaks up those fatty acids and glycerol molecules from each other. Cell is activated by bile salts binding to the enzyme. Describe two other functions of bile salts. So I want you to think now, pause the video, write down on a bit of paper anything that you know, any keywords linked to bile salts. Okay, hopefully you came up with these. So emulsification. So I'd say it plays a role in emulsification and then plays a role in the formation of micelles. I can't be bothered to write the whole sentence out because I'm feeling lazy now. So plays a role in emulsification and in the formation of micelles. And there you go. That's just how you're doing word association. Easy peasy. last part to question 6. Mammals feed their young on milk. Cell digests triglycerides in milk. The ability to produce cell occurred during a gene mutation. Describe how natural selection may have led to all mammals in a population producing cell. So what we want to do here is you should write this down on a revision card and have it memorized off by heart. I'm going to write it in white over here. This is the kind of general process of natural selection. And then what you're going to do is you're going to switch it up for each question to make it apply to the question. So most natural selection questions go like this: variation in a population, right, is caused by mutations. So we know that we got variation in a population is caused by mutations. Beneficial alleles make individuals more likely to survive. You know what they called that, don't you? The catchphrase, survival of the fittest. So beneficial alleles make individuals more likely to survive, whether it's a bigger fin on a shark so it can swim faster, or black and white stripes on the zebra confuse the predators. Okay, whatever the feature is, it could be your nostril hairs, your ear length, whatever. They've all been chosen by natural selection, every single feature of every individual within a population. General features are chosen by natural selection or developed by natural selection. So once you've got this beneficial allele and you're more likely to survive, what are you more likely to do? You're more likely to get down. Yeah, you're more likely to reproduce. Okay, so we got some reproduce. That was bad, wasn't it? I should have said, yeah, breed, reproduce, have offspring, whatever. Have offspring. Oh, that's really rubbish, isn't it? Pass on the beneficial allele to the offspring. Notice at A level, we want to really ram home, pause, no diddy. We want to ram home the allele, the word allele. That's what's different to the GCSE version of this. And then we want to say, over many generations, over many generations, the frequency of this allele increases in the population. I got some spelling mistakes there, but what are you going to say? Called it beneficial. Beneficial. Oh my gosh, I can't even read that now. Hey, yeah, I'm just gonna have to go with that, guys. You're gonna have to allow on the spelling mistakes because when it goes white, I can't even see what I'm typing. So this is like a general one. Okay, one of the key things is increase in the frequency of this allele in the population. That's an allele when you must say that. All I would say is now apply this to this, because this is talked about triglycerides. So I would now make this specific to triglycerides to say, okay, so the beneficial allele we've been told gives you the cell enzyme, means you're more likely to hydrolyze triglycerides into fatty acids and glycerol. These individuals had more energy, more access to more ATP, and anything else that we can get from triglycerides. So I'd say more energy, more ATP, greater muscle contraction, whatever, run faster in the mammals, produce more heat to regulate their body temperature. So things like that. You want to apply it specifically to this question. And that made them more likely to survive, more likely to reproduce, more likely to pass on the beneficial allele to their offspring. And over time, this increased the frequency of the allele in the population. So I'd say a couple of the marks you want to make specific to the question. But first of all, you've got to memorize that process of natural selection on the left. Anyway, that's how you answer that question. Just add in the bits about triglycerides and how they give you more ATP and more heat is released and allows you, mammals specifically they talked about here, able to regulate their body temperature or run faster, contract their muscles more, whatever. Something about the benefit of ATP. And here we go. 7.1, figure 4 shows the mass of DNA present in a group of healthy cells. We've got the number of cells with this mass of DNA, and then the mass of DNA in the cell in arbitrary units. So this graph can be a little bit difficult to read, but essentially all of these cells here and the most of the cells, look, the highest number of cells is the number of cells have a DNA mass of 1. And then there's a load of cells with a kind of DNA mass in between 1 and 2. And then some cells with a mass of DNA that is double the mass of the DNA in 1. Use your knowledge of the cell cycle to explain the results shown in figure 4. Okay, now what we can see here is that cells with a DNA mass of 1, which is the majority of the cells, or a large amount of the cells, not the majority, but the most of the cells have a DNA mass of 1. This must be early in the interphase, before the DNA has started replicating. I can't spell interphase, that's disgusting. Then we need to make a comment about the cells with a mass of 2. Remember, this is a 3 marker. And then we need to make a comment about the cells with a DNA mass in between 1 and 2. So I'd say these cells which have a DNA mass of 2, cells with a DNA mass of 2, what we can say is they're in the G2 phase, or we can say they are undergoing mitosis. The chromosome must be made of 2 chromatids. So we'd say each chromosome at this stage, each chromosome is made of 2 chromatids. So essentially, they've got to the point where, the chromosomes have gone from being like this to having 2 identical sister chromatomes, right? That would explain why their mass has doubled. So we're talking early, what do we think? Prophase of mitosis, something like that. And then what I would say is there's no um, there are no cells with half the DNA of an ordinary cell. And so there are no cells. So we know that it is mitosis and not meiosis. That's another comment we could make. I'm kind of running out of room. What I would say is we've made a comment about here, and we've made a comment for this one about here. We now need to really make our third comment about here. What does this tell us here? I'm going to write the comment over here because I'm running out of room. So if we look at that section in the middle, we would say, yeah, the mass of the DNA. So let's go mass of, woo, can't spell, mass of DNA between 1 and 2 indicates a DNA being replicated, basically DNA replication is occurring. And to me, this would suggest cells in the S phase, the synthesis phase, which is where the DNA is replicated. And that's what would be indicated there, because these guys have got greater DNA than the normal cell. And yeah, there we go. These ones have got double bubble, and there's none over here. So there's no cells with zero point 0.5 the cell DNA, and that would mean that's what we would expect with meiosis, with haploid cells. So another comment I could make: no cells have half the DNA. Oh my God, half the DNA. This means no meiosis, right? No haploid cells. So yeah, yeah. So I think basically we've got kind of 4 marking points for 3 things. 7.2 suggests one way figure 4 be different if these were tumor cells. So what is it that tumor cells do? Tumor cells, we've got uncontrollable mitosis, haven't we? Uncontrollable cell division. So we would expect the bars to be higher with tumor cells. Fewer cells with a mass of 1, more cells between 1 and 2, showing that we've got faster uncontrolled cell division. Yeah. So more cells would be undergoing division. So we'd expect more cells not to be in the early interphase, but to be in the dividing phase. So where they've got like undergoing mitosis. So faster cell division, more uncontrollable. I can't even spell uncontrollable mitosis. And because that's the case, I think we'd see a greater number of cells with double the DNA. So we would see with a mass of 2. That would indicate more cells in the prophase or more cells undergoing mitosis where they're going to have sister chromatids, so double the DNA. And that's because tumor cells have uncontrollable mitosis. We'd actually have less with a mass of 1 because more of the cells, so a higher proportion of the cells would be undergoing mitosis. Describe the behavior of chromosomes in the prophase and metaphase. This is just a basic content one. So if you remember the prophase, we're looking to see randomly kind of scrawled. The chromosomes haven't lined up in the center yet or sort of got organized yet. So I'd say they're shortened, fattened, condensed, randomly arranged. So randomly arranged. The question implies we're talking about chromosomes, so I don't have to say randomly arranged chromosomes, but randomly arranged, shorter. Oh my days! Visible under a light microscope. Remember, shorter, fatter, uh, condensed is kind of like what I would say. The metaphase, the chromosomes, we'd expect them, I think of the metaphase as the meeting phase, the word meet. So they've all met along the equator, right? So we'd probably see chromosomes lined up along the equator. So chromosomes lined up on equator, attaching to spindle fibers, all of that business. Right, there we go. We only need to say one of each. Ba, ba, ba. Oh my days, we've got a maths question here. So what we're going to do here, let's go through. 7.4, during anaphase, the spindle exerts 3 times 10 to the minus 11 newtons of force. So this is F. I can already see there's an equation here, so I'm looking to find F, P, and V. So we've got the newtons of force there. That's F on each chromatid. This force generates 6 times 10 to the minus 19 watts of power. So that is P in that equation there. Calculate the speed of movement. What's the unit in nanometers per second of one chromatid during the anaphase using the following equation. Power F V. The thing to note here, and you might miss this, this will cost you a mark, is that the units we're going to calculate, the V we're going to calculate is in meters a second, and they want the V in nanometers a second. So there will be a conversion to make at the end. Let's go ahead and do this. Now, if you are somebody who likes triangles to help you with rearranging, you would put the P at the top and F times the V at the bottom, put a dividing line there. We want to find V because they've asked us to calculate speed, which is kind of the same as velocity. All right, physicists, I know you're screaming about scalar and vectors, but whatever. Right, pressure divided by force gives me velocity. So the pressure was 6 times 10 to the minus 19. I want to divide that by 3 times 10 to the minus 11. Let me see what that's going to give me. That's going to give me some small ass number. So let's do that. That's 6 times 10 to the minus 19 divided by, no, not take away, divided by 3 times 10 to the minus 11. And that gives me like 1 over whatever the hell that is, 50 million or something, which is 2 times 10 to the minus 8. All right, whatever that is. Yeah, I'm not going to sit here and worry about it. 2 times 10 to the minus 8. And that is going to pick me up. I think that's going to get me 1 mark there for working that out. That is the velocity in meters per second. What I need to do now is convert that into nanometers per second. Now, what's unusual is when you go from a bigger unit to a smaller unit, it's counterintuitive. That means goes against the way our brain normally thinks, a bit like the left side and right side of the heart diagram, when you kind of think when you're going to a smaller unit, you multiply. Just think about if you've got to ask yourself, if you get confused, do I multiply or do I divide with these things? Think about your height. You might be 1.7 meters, 1.6 meters. If someone asks you in centimeters, which is a smaller unit, you're like 160 centimeters or 170 centimeters. So as we go down with conversions, we multiply and get a bigger number. As I forgot the micrometers and nanometers. So we're timesing by 1,000, by 1,000, by 1,000. You're basically timesing by 1,000,000,000 to get these guys there. So timesing, what am I waffling about? So let's times that up by 1,000,000,000. If you don't feel confident with smashing out the calculator, do it times by 1,000, times by 1,000, times by 1,000. And the answer is 20 nanometers per second. You're kind of wiping off three zeros each time. Okay, question eight. A student investigated a method of estimating the concentration of protein in a solution by using a measure of the density of the solutions. The copper sulfate solutions have different concentration of known densities, so they can be used to measure the density of other solutions. The student prepared a dilution series of a copper sulfate solution. Complete the table by giving all the headings, units, volumes required to make 30 centimeter cubed of concentration of copper sulfate solution. Bum, 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 bum. Um, Shown. Okay. So what have we got to do here? 100 grams, kilograms copper sulfate solution. Um, it tells us here. Okay, let's get into this. We need to make 30 centimeter cube. That's our volume. It has to be 30 centimeter cube total volume. They're telling us the concentration has to be 75 in 1,000 grams, right? We need 75 in 1,000 grams. Our problem is that we've been given like a stock solution of 100 grams in 100 grams of copper sulfate in 1,000 grams of solution. So we've got a kind of, this is a 7.5% solution we want, and we've been given a 10% solution. So if you've only got 30 centimeters to play with, how are we going to divide this up? And we're going to use centimeter cube for both of these. So that's probably going to get us a mark in the headings. And we need to divide this up. Well, I want to dilute this 100 grams down to its only 7.5% from 10%. So I need to dilute it by basically 25%. Yeah, I'm going from 10%. I'm going from 10. I'm going from 100 to 75. So to me, that means I'm making it a quarter weaker. I need to make this 100 to 1,000 a quarter weaker, and I've only got 30 centimeters to play with. So I'm going to do 30 centimeter cubed. It's up to you how you do this. You can either times it by 75%, because I only want 75% of the copper sulfate of that, or you could do it the other way around, and I'm going to times it by 0.25. 25%, that's like, and that's going to give me 7.5 centimeters. So I want to whack in 7.5 centimeters of water, and that's going to leave me with 22.5 centimeter cubed of this 100-gram one. So essentially what I'm doing is 75% of the 30 centimeters needs to be this 100 grams, and 7.5% needs to be water. Why? I'm diluting the whole thing. I'm diluting this by 25%. Okay, and if that makes sense, you're a genius. So you're going to get a mark for the headings. You're going to get a mark for the volumes. Right, let's move down. Okay, sorry about that interruption. So question 8.2, table 4 shows the densities of the dilution series of the copper sulfate solution. So here you got the different densities, and this is our standardized copper sulfate solution. We're going to use this to estimate the concentration of unknown protein solutions. So figure 5 shows the densities of protein solutions of different concentration. So for example, if it was 20% protein solution, would come up here, smash along here, would check out the density of that protein solution, and then go and well, yeah, you get the idea. And then we'd be able to compare it to the copper sulfate solution. What am I waffling about? Okay, let's do this. The student put 1 drop, 10% of protein solution into each copper sulfate solution shown in table 4. So basically, they put 1 drop of a 10% protein solution into each one of these. Let's zing down and read the rest of the question. Using figure 5, he predicted the drop would sink in the 0 and 25 gram per kilogram copper sulfate solutions, and it would float in the 50, 75, and 100 gram copper sulfate solutions. Give the density of the 10% protein solution. So we've got to show how we can use the graph, and explain why the student predicted that the drop would sink in 25 gram per kilo copper sulfate. Okay, let's do that. So the first thing is to give the density of it and then compare it to the 25 gram copper sulfate. So they asked us to give the density of the 10% protein solution. So this is just first of all, we want to work out how much each one of these little squares is worth, right? Each square, what is it? The value is, oh my days, 1.01 to 1.03. That means we're going up by 0.02. And then we divide that by 10 squares. And we're going to find out how much each square is worth. 0.02 divided by 10 equals, each square is worth 0.0, oh my days, 0.02. Oh my good gosh. So this is, all right, we worked that out. We worked that out. Right, we're looking at the 10%. So you go up here, come to this black line here, then draw it across. Bang, it's one down from one, it's one square down from 1.03. That must be 1.02. So 1.022, 1.024, 1.026, 1.028. So we're looking at a density of 1.028, and that is gram per centimeter cubed. So remember that. We're going to go down and put that in. 1.028. So it has a density of 1.028 gram per centimeter cubed, and that would get us a mark. Now, what's the explanation as to why that would sink? Let's go up to the 25. And no, that's the protein. Where's the table? Aha! Concentration of copper sulfate solution when it's 25. Concentration of the copper sulfate solution at 25 is only 1.014. So let's go back down and write our answer. We got the answer right there. So what it is, this was 1.028. I'm struggling to remember. Explanation, the copper sulfate solution at 25G is 1.014. So the protein solution has a higher density, so would sink, i.e. the protein solution has a density of 1.028, which is greater than 1.014 of the copper sulfate, so the protein solution will sink in the copper sulfate. The dude was correct. Well done. Now let's boom down here. State the range of possible concentrations of a protein solution that sinks in 75. So it sinks in 75 and it's more dense than 75 and floats in 100. So what's the minimum concentration and what's the max concentration? So, woo. So 1.048 to 1.065. Let's go down. So if it's going to sink in 1.048, it's going to have to be like of that density or greater. And then if it's going to float in this, it mustn't be greater than 1.065. So let's check the range. So the range, let's find 1.048 density here. So we go 1.048 is there. Let's bash along to here. Oh my days. Could I draw a line any more bent? And then down there. I've got to work out what that is. So each one of these is worth 0.5. If you're wondering how I know that, that is 5 over, there's 10 squares in the range and the value is 5. So it's 0.5. So let's work that out. That is 15, 15.5, 16, 16.5 is the minimum concentration of the protein. And then we're looking at the range of the protein. And what was the other one? 1.065. So that's 1.06. We're going to go one up. That is 1.065. Smack along to the black line. Come down. Oh my days. This looks like 20.5, 22? No. What is that? 21? My gosh, I've completely lost my ability to think. So So we need to find 1.065. So if that's 1.05 and that's 1.07, here is 1.06 and then 1.065. What did we say each of these was worth? Um, zero point 0.2. So 1.065 is going to be halfway between 1.07 and 1.06. So it's going to be there. Oh my days, that's so bent. Yeah, that looks about right. And then if I come down here, what's our reading here? Each one of these was zero point 0.5, so 20, 1, 21.5, 22. Right, so we're looking at our range is 16.5% to 22%. And here we go. Let's write that in on the answer. So, bish bash bosh. So the minimum concentration is going to be 16.5 percent. So this was just a graph reading exercise, and that is 22%. Boy, I could have made that easier, but there we go. Okay, lovely. Okay, 8.4. Blood donation involves healthy donors giving blood that can be used to treat hospital patients. When donors arrive, the hemoglobin concentration of their blood is tested. A sample of each donor's blood is added to the copper sulfate solution to determine whether the hemoglobin concentration is high enough to donate. Error sometimes occurs with this test. Tom has a concentration of hemoglobin high enough to donate. Lucy has a concentration of hemoglobin too low to donate. Evaluate the consequences of the errors occurring when Tom and Lucy's blood samples are tested. Okay, let's zing down so we can see here it's worth 3 marks. Okay, so Tom does have enough hemoglobin, but let's imagine the test screws up, right? It goes wrong. And it comes out that Tom doesn't have it. It looks like he doesn't have enough hemoglobin. Um, what does this mean? What's the consequences? It means healthy um, blood will not be available, i.e., there's a reduction in the blood available to donate, is a better way to say it. A reduction in the blood available to donate. So, so patients might not get treatment. So if they mess up and it comes out that Tom's hemoglobin is too low, some patients might miss out on some really good quality blood with lots of hemoglobin. Also, this could cause anxiety or stress, whoopsie, to Tom, right? Because all of a sudden he's gone through his life thinking he's healthy, and boom, the doctor says, sorry, you haven't got enough hemoglobin in your blood to be a blood donor. And he goes away stressing about the lack of hemoglobin in his blood. Now, what are the consequences for Lucy's blood? Maybe her blood, which we know is too low in hemoglobin to donate to patients, because it won't work effectively at carrying oxygen. But if it comes out positive, like that her blood does have hemoglobin, when it doesn't, then a load of patients may receive blood. So patients may receive blood um, with not enough hemoglobin. So that's not healthy for the patients. What else? Lucy may believe her blood is okay and not find out that her blood is low in hemoglobin. Now, I'm being lazy and I'm just using HB for hemoglobin. You can't really use abbreviations unless they tell you that you can, so better not to do that. Um, yeah, and also another thing, if she donates blood, she may be left with not enough hemoglobin to carry oxygen. This could be negative for her health. So basically, if she doesn't have enough oxygen, this could be really negative for her health, right? She's given away blood. She thinks she's got more hemoglobin than she does, and this could mash her health up. Okay, so there's 3 points there for her as well as those three. Let's move on to the next question. Bosh. Scientists dissected gills from several species of fish. They recorded the mass of the whole fish, the total number of gill filaments, the mean length of one filament, the mean number of lamellae per millimeter, the mean surface area of one lamellae. Whee! It was not possible for scientists to measure the length of every filament and the surface area of every lamellae. Of course not. That'll take them years. Suggest how they collected the data to give a reliable mean for these variables. Well, if you want a reliable mean, this is really just testing you on what you need for a reliable mean. We need um, they should use a large sample size. So they should collect lots of fish of whatever species. Let's say they're going to look at some cod, the fish cod. They should get thousands of cod, obviously, to make it better, a nice large sample size, and then randomly choose fish from the sample, right? So they want to do, this is known as random sampling. And that way, if you've got 1,000 cod, you don't want to be checking every single cod's gills and measuring them all. You want to take a little sample. So maybe you want to take 10 or 15 cod and calculate the mean from that. But you want to make sure you select them randomly so there's no bias involved with your selection process. And that will give you a reliable mean. Okay, let's move on. 9.2. For these measurements, the scientists calculate the total surface area of the gas exchange surface on the gills of each fish species. Calculate the total surface area. So we're being asked to calculate here. So it's a maths question, obviously. Calculate the total surface area of the gills of a fish with the following measurements. Give your answer in millimeters squared and to the appropriate number of significant figures, show you're working. Right, we've got 595 gills, right? The mean length of one gill filament is 2.86 millimeters. The mean number of lamellae is per millimeter is 16, right? Lamellae are like these disc-shaped things that give a larger surface area to the gill filaments. So these are lamellae, these things here. I can never spell that right. And this is gill filament. Okay, coolio. Right, now what they're saying is that if you only look at if we only look at a millimeter, if we look at just say 1 millimeter for these gill filaments, what we find is there are 16 lamellae in 1 millimeter. The mean surface area of a lamellae is 0.66. Give your answer in millimeter and the appropriate number of significant figures. We want to calculate the total surface area. Right, well, that means we've got 595 of these bad boys. So let's get down to this. So, choose a different color, Tom. Right, we've got 595 filaments, and each one has a length of 2.86. So I'm going to find out the total length of all my gill filaments in millimeters. So total length is 596. What did I say? 595? Was it 595? Yeah, 595 times 2.86. Smash that in the calculator, and we should get 1,701.7. It's always good to write out your working. That's the total length. And the reason is that often on the mark scheme, you get 1 mark for one of these numbers appearing. So if the examiner sees that, they may go, ooh, give them a mark. So we've now got the total length. We know there are 16 per millimeter. So I've got 1,701.7 millimeters, and I've got 16 in each one of those. So guess what? I need to times by 16, and this will give me the total number of lamellae. And so let's times that by 16, and that is going to give me 27,227. So equals 27,227.2 lamellae. Yeah, right. You get the idea. That's the total number of lamellae. So now I know how many discs there are altogether. The surface area of each lamellae is 0.66. So what's the total surface area? Well, it's smaller than the number of lamellae because I need to times by 0.66. So you might get confused and think, I haven't done any division. You don't need to here. Okay, times by 0.66 because we're working out the total area. And that equals an area of 17,969.952, and that is millimeter squared. Now, it did say give to an appropriate number of significant figures, so I might round this to 2 sig because can take this up to 18,000. That's probably best. Or you might want to write this as 17,970,000 by rounding that up because of these. And that might be a nice thing as well. But any of these will get you the mark. So I'm going with 18,000 or 1.8 times 10 to the whatever the hell that is for 1.8 times 10 to the 4 or 18,000 millimeters squared. There we go. Now they'll probably give you a mark for any of these numbers, like calculating the total length or the total number of lamellae or the surface area before rounding, and then bosh, you get 2 marks. All right, let's move on quickly. 9.3, the table 5 shows the scientist data for 2 species of fish. I've got Opsanus tau, and it has a mean mass of 305 and a mean surface area of 46,100 for its gills. Then I have Trachurus, and that has a mass of 250, so it's lighter, but it has, a much bigger surface area. It's over 5 times bigger than the Opsanus tau. One of these species spends most of its time not moving, just chilling out, being fat and lazy like myself, waiting to catch passing prey. The other species is very active, hunting mobile prey. That means prey that are moving. So suggest which species is the very active species. Explain your answer. Well, it's pretty obvious, right? It's got to be this Trachurus, however we say that. Trachurus. Can't spell even if the spelling's in front of me. Why? So we're going to get a mark for that. And then the explanation, it's got a large surface area to volume ratio of its gills. So large SAV of gills. Large means surface area, means it can, I don't know, absorb. Oh, this is disgusting. Absorb oxygen into the blood quickly for aerobic respiration to produce ATP for muscle contraction. Why does it want to contract its muscles? It's moving fast to catch the prey, needs loads of ATP, going to need oxygen to do the respiration to make the ATP. Okay, so large surface area of the gills. So, or we can just say large, large mean surface area of the gills means it can absorb oxygen quickly, or oxygen will diffuse. Maybe they want me to use the word diffuse, means it can absorb oxygen quickly. Oxygen diffuses quickly into the blood. So maybe I get a bit paranoid and I think, yeah, let's smash that in there and make sure they don't have something like, oh, you didn't mention diffusion, right? And there we go. I'm ticking everywhere. Complete table 6 to show the phylogenetic. Excuse me, I've got such a bad chest at the moment. Complete table 6 to show the phylogenetic classification of these 2 species. So you've got to know your taxons, and you might think, oh, we're starting with domains. We're not, because you can see Animalia is over there. So our taxon, what is Animalia? It's one of the kingdoms. And if you want to remember this, some people say kings, play, cards, on, fat, girl, stomachs. Not very nice, that one. How about king prawn curry or fat greasy sausages? Well, there's actually loads of ways to make it. So the next one is kingdom, phylum. Is it 2 L's? Don't know. Class, order, family, genus, species. Lovely. So we're going to get some marks for doing that. Hopefully get at least 1 mark for getting all the taxons right. And then over here, the kingdom. Well, they're both fish. They're both part of the animal kingdom. So they're both fish. So it's going to be Animalia again. And then chordata. Do they have a spinal cord? Yeah, fish have a spinal cord. That's basically a bit like saying vertebrae. So they're both chordata. You're not expected to know Animalia or chordata off by heart, but you should be able to apply your brains to work it out from the fact they're both fish. They're both in the animal kingdom. If that one's a chordata, this one's a chordata. And look, they share the same class and it's hierarchical. So if they share the same class, they must share the same phylum. They must share the same kingdom. So you can work it out like that. And then we start to diverge here genetically once we get to the order. And then obviously the family is going to be different if the order's different. And then the genus, how do we know the genus? We work it out from the binomial system. So Opsanus tau. Tau is the species and Opsanus is the genus. Trachurus is the genus. You must spell it with a capital letter, otherwise they won't give you the mark for that one. I know I've kind of like not spelt the others with capitals, but this is vital because it's part of the binomial system, species lowercase and capital for genus. Right, that is the end of question 9. Just one more question to go. Yippee yippee do. Okay, question 10, 10.1. I've got a terrible cold, peeps. I'm going to come out sounding very nasal like this. Okay, outline the similarities in and the differences between the structures of DNA and RNA molecules. If I'm in an exam, I want to draw these out so I can like definitely work it out. Oh my days, that is the worst DNA. Yeah, anyway. So I'm looking at there's my DNA, there's my RNA, and then we've got to think about they're both made of nucleotides, aren't they? So, oh my gosh, have you seen a nucleotide that bad? Not sure I have. Right, let's get into it. Let's write them down, these differences and similarities. So, start with the similarities. Both are polymers made of nucleotides, right? Um, yeah, i.e., both contain phosphate groups, um, pentose sugars, both deoxyribose and ribose are pentose sugars, and adenine, what else? Guanine and cytosine bases. So there's some three similarities right there. Oh my days. So I would say that is like 3 marks straight away. Oh, what kind of bonds connect the nucleotides together in DNA and RNA? So both contain phosphodiester bonds. Nice. I'd say that is another similarity. Pum. So we've got the nitrogenous bases, adenine, guanine, cytosine. That's 1 mark. We've got they're both made of phosphate groups and pentose sugars. I've never seen something called a pentose dizzy sugar. So we want that. And both are polymers made of nucleotides. Both contain phosphate. I think that's enough similarities. Let's get 4 differences, 5 differences. So let's start with the base difference, right? RNA contains uracil, whereas DNA contains thymine. So they don't, all the bases aren't the same. What can we say? We can say RNA contains ribose sugar, whereas what's the sugar in DNA? Whereas DNA contains deoxyribose sugar. There's another difference. This is double-stranded, so again, some of these might not be on the mark scheme, but I'm just going for every single one because in an exam situation, you wouldn't be thinking about exactly the mark. You just want to get everything down. DNA double-stranded, RNA single-stranded. Now, this next one probably doesn't count as a point, but I want to get everything. It's not going to lose me a point, so I'm going to say DNA is longer and RNA is shorter. That's probably not good enough for A level. They probably won't have that on the mark scheme, but just in case, I'm slapping that down there as well. DNA has complementary base pairing, doesn't it, over here between the nitrogenous bases. So DNA has, whoops, no need for a capital, has H bonding or hydrogen bonding between complementary bases. So we've got complementary base pairing in DNA. There's no complementary base pairing in RNA because it's single-stranded. So that leads off of that. I reckon that's about it, you know. Can't really think. I think that's about it. But like I say, there's probably 8 points there. 1 or 2 might not be on the mark scheme. Maybe the shorter and longer isn't on the mark scheme. But you've definitely covered all 6 marks there. And it just helped me by drawing this out. So, you know, you can think of all the things. I mean, really, you know, you've by drawing it out, even that D grade level art, you might remember the phosphodiester bonds or something. Okie dokie. Let's move on anyhow. We're up to the end of the test now. So I believe this is the last question in the test. 10.2, outline the similarities in and the differences, another similarities and difference between the structures of chloroplasts and mitochondria. So let me sketch that out for you. You should all be able to sketch these out because these are going to help you. I know they're crusty little drawings, but these kind of things, imagine if someone said draw a chloroplast. If you can't do that, then do you really know its structure? You don't. So there's my chloroplast. Now, if I was doing mitochondria, what does a mitochondria look like? Oh, I'm so bad at this. I'm not very good at the old mitochondria. All right, you might have to forgive this mitochondria. This mitochondria looks like a monster's mouth. Um, I'm not sure that is exactly what a mitochondria looks like. That is some bogus drawing. Right, what's the similarity? From that, I would say both contain a double membrane. Okay, they've both got an envelope. They're both double membrane bound organelles. Both contain a double membrane, or I can say are double membrane bound. So they're bound in a double membrane. That's a similarity. Let's see, what else? What else? What else? Both have cytoplasm. No, they don't have cytoplasm, do they? Aha, that's a difference. What's the jelly in both? That's a difference. Okay, what's another similarity other than the membrane? Ah, both have ribosomes because endosymbiosis, they were once their own little living things. So they had to make protein, which also means both have their own DNA. Both have their own DNA, and their DNA, probably the same marking point, their DNA is circular. So they have circular DNA. It's still double-stranded because it's DNA. It's not linear, but and it's yeah, sorry, it's circular. It's not linear, it's circular. They join up at the ends. So both have their own DNA. The DNA is circular, both have ribosomes, both contain a double membrane. I think I'm stretching here. I think that's the three. That's the three. Okay, let's move on. Let's go to three. Oh, it's only a four marker. I'm trying to get 6 points. It's only a four marker. So yeah, there's probably less similarities. Well, let's go for the differences. Loads of differences. So what do we call these structures within a chloroplast? So first of all, I'll talk about the jelly because that's the bit that I messed up on. I'm starting to say they both have cytoplasm. So chloroplast has, whoa, has stroma. That's the fluid inside. Whereas the mitochondria has a matrix. That's a direct comparison there. Difference. Mitochondria has cristae. That's where like all its enzymes are. I would say chloroplast. This isn't a direct comparison, but chloroplast has thylakoids. On that note, we could say grana. Whoops, grana. um, or intergranal lamellae. I'm just shoving everything down. That's different, really. Um, so mitochondria's got cristae, chloroplasts have thylakoids, granule or intergranal lamellae. Oh, chloroplasts have starch grains sometimes, don't they? Chloroplasts have starch grains. So I know they've got circular DNA, but this might be my little starch grain over here. Yeah, you're not going to get starch inside a mitochondria. It's glycogen that's well, you're not even going to get glycogen in a mitochondria, but yeah, there's going to be no granules in there, whereas there's starch granules here. Okay, cool. That is going to be at least 4 marks. All right, guys, don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. I'll catch you guys in the next one. Exams are coming, revise hard. Good luck.
You're still here? It's over.